Hi everyone, my name is Ultrashort. I'm a PhD student at the University of Arizona James C. Wyant College of Optical Sciences. I work here with Professor Khan Q on the research of ultra-short pulse fiber lasers and its applications. Let's coming to see what cool stuff our team works on here. Right now I am in our ultra-short fiber laser and nonlinear optics laboratory. Lasers are one of the most important tools in today's science and technology advancement. The main advantages of ultra-short pulse fiber laser systems are that it can be very compact in size, also very reliable, and cheaper compared to other type of pulse laser systems. Semiconductor diode laser pumping technology make it more efficient. Nowadays we can get over 70% efficiency optical to optical and over 40% efficiency from electrical to optical in ytterbium laser systems, for example. The compactness of fiber laser comes from the flexibility of optical fiber so that you can coil it in very small space. Also, the fiber coupled components can be directly spliced to one another, and this technology developed very well. Like, we can find several different types of splicers from the market, including arc fusion splicer and filament type of splicer. Currently, we are focusing on developing ultra-short fiber laser systems that are useful in many different applications, but the spectral region is not covered by the rare earth doped active gain fiber based lasers. Our most recent results were ultra-short all fiber 1300 nanometer and 1700 nanometer laser systems. At 1300 and 1700 nanometer, the water absorption and the scattering loss is very low. Therefore, it's very useful source for deep tissue imaging. The laser pulses are confined very strongly inside the optical fiber because it's a very tight space. Therefore, it's very important to understand what exactly happened to our optical pulses, laser pulses, so that we can resolve the pulse distortion issues by optimizing the system or choosing the right fiber. The master equation that explains pulse evaluation inside the optical fiber is called nonlinear Schrödinger equation, which is derived from Maxwell equations. It is a third-order nonlinear equation, and we solve this equation by numerically to get deeper insight about our systems. In order to get most useful information out of this simulation, we need to model the optical fiber correctly, which means that we need to calculate or measure the dispersion parameters of the optical fiber, nonlinearity parameter, and then Raman response function of this fiber. As you see from here, the simulation results are well matched with the actual experimental results. There are many useful applications that ultra-short fiber lasers are suitable for, from the internet that we use in our daily life to the nonlinear imaging technologies that could be a promising tool to diagnose cancers in early stage. In our microscopy lab, we develop several different types of nonlinear microscopes, as well as use them to study about variety of samples from gem minerals to brain neurons. In general, the microscope is a tool to see structure of tiny little objects by magnifying it with the help of imaging optics. Nonlinear optical microscopy uses nonlinear interactions between light and the object to generate images in contrast. It doesn't require any additional stain to label the structure of the sample, therefore we call it label-free image. Combined with ultra-short fiber lasers, our microscopes are extremely simpler to operate. From this image of pancreas sample, you can see how easy to distinguish between cancerous region and normal healthy region with the nonlinear optical microscopy. Because all these nonlinear processes require very high laser power, it mostly occurs at the focal spot of the imaging system. Therefore, it makes it possible to image the sample in 3D. For example, from here you can see the 3D render of mouse brain sample. You can see the neurons very clearly. These are some representations of our current results. In our microscopy lab, we are developing stimulated Raman microscopy that can detect many different types of bacteria, and we think that it also can detect the coronavirus as well. Our team has members with different background level in optical sciences. We have undergrad students, master students, and PhD students. We learn from each other every day and grow together. I think this is one of the most important things that academic and research environment must have. We would like to acknowledge those who support us to make all this interesting research going. Thank you very much, Quentin Salter from the Office of Naval Research, Directed Energy Ultrashort Pulse Laser Program, as well as NIH and NSF. Thank you very much for watching this video. Stay well during this pandemic time. Wear your mask. Keep the 6 feet distance. Thank you.